Hi, I'm Jiao Yan from South China University of Technology. Today is my great honor to be here to share some knowledge about biochemistry. Biochemistry actually is an evolving science, as we all know as the study of chemistry of life processes. These processes entail the interplay of two different classes of molecules, like large molecules such as proteins and nucleic acids, referred to as biological macromolecules, and low molecule weight molecules such as glucose and glycerol, referred to as metabolites, that are chemically transformed in biological processes. Note that life on Earth began approximately 3.5 billion years ago, whereas human beings emerged quite recently. On the basis of their biological or biochemical characteristics, the diverse organisms of the modern world can be divided into three fundamental groups called domains, including eukaryotes, bacteria, and archaea. Modern cells contain high concentrations of RNAs, small molecules and large molecules aggregates that are found only in traces outside the cell. For example, a cell of the E. coli contains millions of molecules, representing some 3,000 to 6,000 different compounds. And a different animal cell may also contain 100,000 different types of molecules. All modern organisms are based on the same morphological unit we call the cell. There are two major classifications of cells, like eukaryotes, which have membrane-enclosed nucleus encapsulating their DNA, and prokaryotes, which lack a nucleus. Okay, so prokaryotes comprising the rhesus type of bacteria have relatively simple structure and are almost all unicellular. While eukaryotes are vastly more complex than prokaryotes. Also here, viruses are much simpler entities than cells that are not classified as living because they lack the metabolized apparatus to reproduce outside their host cells. So prokaryotes range in size from 1 to 10 micrometer and have one of three basic shapes, including spherical, rod-like, and helicoidal coiled. Eukaryote cells are generally 10 to 100 micrometer in diameter and thus have 1,000 to 1,000,000 times the volume of typical prokaryotes. It's not size, however, but a profusion of membrane-enclosed organelles that's best characteristic eukaryote cells, and we will introduce some of them. Okay, so in addition to a nucleus, eukaryotes have an endoplasmic reticulum, the site of the synthesis of monocellular components, some of which are subsequently modified in the Golgi apparatus. The bulk of aerobic metabolism takes place in mitochondria in almost all eukaryotes. And the photosynthesis cells contain chloroplasts, which convert the energy of the sun's rays to chemical energy. Other organelles, such as lysosomes and peroxisomes, perform specialized functions. Vacuoles, which are more prominent in plant than in animal cells, usually function as storage depots. The cytosol is organized by the cytoskeleton, an extensive area of filaments that also gives the cell its shape and the ability to move. The structure of DNA powerfully illustrates a basic principle common to all biological macromolecules: the innate relation between structure and function. The remarkable properties of this chemical substance allow it to function as a very efficient and robust vehicle for storing information. DNA is a linear polymer made up of 4 different types of monomers. It has a fixed backbone from which produce variable substitutes, referred to as bases. The backbone is built of repeating sugar phosphate units, joined to each deoxyribose as one of 4 possible bases. Adenine, short for A, cytosine, short for C, guanine, short for G, and thymine, short for T. Most DNA molecules consist of not 1 but 2 strands. In 1953, James Watt and Frank Crick proposed a 3-dimensional structure for DNA molecules. This structure is a double helix, and it has 2 properties of central importance to the role of DNA as the hereditary material. First, the structure is compatible with any sequence of bases. While the bases are distinct in structure, the base pairs have essentially the same shape and thus fit equally well into the center of the double helical structure of any sequences. And second, because of the base pairing, the sequences of bases along one strand completely determines the sequence along the other strand. Thus, if the DNA double helix is separated into 2 single strands, each strand can act as a template for the generation of its partner strand through specific base pair formation. The 3-dimensional structure of DNA beautifully illustrates the close connection between molecule form and function. What forces cause the 2 strands of DNA to bind to each other? To analyze this binding reaction, we must consider several factors: the types of interactions and bonds in biochemical systems, and the energetic favorability of the reaction. We must also consider the influence of the solution conditions, in particular the consequences of acid-base reactions. Covalent and non-covalent bonds are important for the structure and stability of biological molecules. A covalent bond is formed by the shaping of a pair of electrons between adjacent atoms. A typical carbon-carbon covalent bonds have a bond length of 1.54 angstrom, and the bond energy is between 347 to 355 kilojoule per mole. The carbon-oxygen double bonds are even stronger than carbon-carbon single bonds and are somewhat shorter. Interatomic distances and bond lengths are usually measured in angstrom units. Several energy units are in common use, like 1 joule, short for G, as the amount of energy required to remove 1 meter against a force of 1 Newton. And 1 calorie as the amount of energy required to raise the temperature of 1 gram of water 1 degree Celsius. 1 calorie is equal to 4.18 joules. 1 joule is equal to 0.239 calorie. For both molecules, more than 1 pattern for covalent bonding can be written. For example, adenine can be written in 2 nearly equivalent ways called renaissance structures. The observed bond length of 1.4 angstrom is between that expected for a carbon-carbon single bond and a carbon-carbon double bond. A molecule that can be written as several renaissance structures approximately equal energies has greater stability than does a molecule without multiple renaissance structures. Okay, so that's all for today. We'll continue next courses. Thank you.